Hello, my name is Gonzalez, and today I'm going to be discussing some techniques for locating snake oviposition sites. The issue here is that there is not a highly effective method that wildlife researchers can use to locate snake oviposition sites, especially for secretive species that conceal their nesting sites. After a thorough literature review, I have found that there are significant drawbacks to all of the techniques that have been used for snake oviposition site location. This can be problematic in a few key ways. For one, it can make it difficult to implement management actions. For example, we have seen this in the case of Boiga irregularis, the brown tree snake. It can also make it difficult to inform conservation actions for native species. Moving along to some of the techniques that have been attempted for oviposition site location, the first step is systematic surveys. This method requires a trained researcher to look for oviposition sites by eye in ideal nesting habitat. The researcher looks for distinct signs of nesting or searches for nests near material that is associated with the oviposition sites of the species of interest. This technique has been successful in locating nests in at least 2 species with distinct nesting site preferences. Also, this technique is relatively cheap and does not require costly equipment or technology. However, this method is highly dependent upon the human individual carrying out the surveys. As some observers may be more or less successful in locating nests than others, there is significant potential for bias and human error. This technique is also generally carried out near known nesting sites. So for species where nesting sites or ideal nesting habitat are completely unknown, this method is not very reliable. Observational studies of this nature may also be labor-intensive for the research team, and researchers may not be able to use this technique in hazardous or difficult terrains. The second technique is radiotelemetry. This involves radio transmitters being implanted into gravid female snakes of the species of interest. The snakes are then released at the site of capture and tracked multiple times per day until oviposition has occurred. In some cases, snakes are tracked and located while they're in the process of oviposition. In cases wherein the snake has oviposited in between observations, a general idea of the nest location is obtained, and the area can be searched further in order to locate the specific site. This technique increases the probability of locating nests when compared with other methods, and it is very reliable for locating oviposition sites in species wherein the process is prolonged or that engage in some sort of nest protection or guarding. Radiotelemetry has also been used with success in numerous species with a wide variety of nesting habits, from striped whip snakes to rough green snakes. However, this method is highly labor-intensive and requires locating snakes on foot numerous times per day. As a result, it may not be appropriate for use in terrains that are difficult to navigate. Further, as snakes must be located numerous times per day, this method can be very time-consuming. Radio transmitters are also very costly, so this technique may be limited in terms of the number of snakes that can be tracked. And finally, as transmitters must be implanted or attached to the snake, this method can be highly stressful to the animal. Finally, we have detection dogs. Detection dogs are trained to detect specific organisms or materials via a target scent. The dog is then brought into the field, where it locates the target scent and alerts the research team. Dogs detect targets more efficiently than human surveyors are able to. Dogs are also ideal for detecting targets in areas that are obscured from vision, which matches the criteria for many snake oviposition sites. Dogs can also locate residual scents, so old or inactive oviposition sites can be located if the research team so desires. However, detector dogs may give false alerts, wherein the target was not actually located. Dogs may also generalize scents, so studies that aim to locate oviposition sites for a specific species may not be the best suited for detector dogs. Training detector dogs is also a very rigorous and time-consuming process, and dogs may not be able to safely traverse certain field sites or terrains, which is another consideration that must be taken into account. Moving on to recommendations, of all the methods examined, radiotelemetry seems to result in the most success for locating snake oviposition sites. While it is a costly and labor-intensive method, radiotelemetry can offer significant insights into snake nesting habits, particularly for those species wherein background information is highly limited or even completely unavailable. One potential modification to this technique involves the surgical placement of a miniature radio transmitter into the oviduct of gravid females. When oviposition occurs, this transmitter is able to be expelled along with the eggs, or young in the case of live bearers, enabling researchers to locate the precise site of oviposition. This is a technique that has been used in fisheries with northern pike to locate critical spawning habitat, and it has also been used with northern long-necked turtles to locate nests that researchers were previously completely unable to find. To my knowledge, this is not a technique that has been extensively tested in snakes. However, it seems like it has a high potential for success, and may be worth exploring further, as it could potentially reduce the very labor-intensive nature of the unmodified radiotelemetry method, as researchers would no longer have to go out into the field and track these females multiple times per day. However, this modification would result in a heightened degree of stress to the animal, and would likely be very costly, so in order to reduce those impacts, it may be beneficial to limit the usage of this technique to cases wherein background information on nesting habits is either completely unavailable or very limited. Further, this modification may not be suited to highly sensitive species, as it may potentially reduce reproductive output when used. So, for species that are endangered or otherwise highly sensitive, it may be best to stick to systematic surveys or detection dogs.